Okay, my friends, Roger once again, Mudfossil University. I've been talking over and over and over about the microbiome. And when I normally talk about that, I talk about bacteria and what are the bacteria that live in the microbiome. Well, guess what? I basically missed viruses because they're now calling it the virome, which is basically the microbiome. This is the microvirome, which is the viruses that live in there. And I guess there's a ton of them. I really didn't consider those, to be perfectly honest with you. But now they are like on the edge of almost alive versus the ribosomes and the enzymes, which are completely not alive. These things seem to be on the edge of life, basically, is the way they describe them now. And viruses, that is. And I want to just show you the main takeaway that we want to think about as we start. Because they're talking about the extent of the virome diversity in the gut remains poorly. They don't know anything about it, let's put it that way. Viruses residing in the human gut are thought to act as a key modulator. They change and play around with the gut microbiome through their interaction with bacteria. That's what I was missing, the interaction with bacteria. And of course the host immune system, which is pretty much based around enzymes. That was my feeling in the first. Now, what do the viruses do? They do basically the same thing as the bacteria. I would think they create some form of a chemical interaction with the body, whether it's an enzyme, whatever you want to call it. It's got to be some kind of chemistry. So the viruses, I would say, are doing a very similar thing to what the bacteria do. Let's go a little further with this. They may influence the structure and function of the bacterial community through facilitation of horizontal gene transfer. We're going to look into that in a minute. Nutrient recycling, that we understand because it's breaking down and bringing back stuff. That's the enzyme stuff. Regulation of bacterial virulence. So they're like maybe assassins. They go out and they kill extra bacteria or something. I don't know. Because that's what regulation of bacterial virulence means. It keeps it down. Because you're going to have these bad bacteria, and you got them in you right now. But something is keeping them from becoming, the key factor to invade you. No. So the regulation, that's the key, regulation. No. And a gain of antibacterial resistance. So you're getting some more resistance to these bacteria that you're interacting with. Somehow from these viruses, they could be the programmers. That's what's going on. I got to think about that. They could be the programmers of the bacteria. Because the bacteria are programmed. They're just programmed little creatures. Wow. This has got to go a little deeper here now. All right. Furthermore, viruses play a direct and indirect role in interactions between the human host and a bacterial community. They're the programmers. And they have been shown to exhibit temporal stability as high as that of their bacterial hosts. Let's take this thing one step at a time. All right. I'm going to drag you through this with me. I'm just starting to think about this, to be perfectly honest with you. I hadn't given it any thought whatsoever. I know the DNA in the bacteria comes from somewhere. Where does that come from? How did it get it programmed? There's thousands and thousands of species. Now we're talking about a virus which is not really alive like a bacteria is alive per se. It apparently seems to be able to get inside of things and reprogram them. So it is basically a reprogrammer. All right, viruses are on the edge of being alive. They are alive-ish to be able to invade other cells, which bacteria are single-celled creatures. Now, whether they invade just these cells, every other, well, we know they invade, viruses invade all cells. We know that because COVID and everything else. Now, viruses have to be the programmers. They're the ones that come in and reprogram bacteria. Because every time you get attacked by something new, a novel, new disease, a new, even virus, we have to have new bacteria. Maybe we have to have new viruses to attack those viruses. I don't know. Now it's sort of taking on a little cloudiness. I was pretty well locked in on these here, creating the ribosomes, and the ribosomes being these little things right here, these tiny little balls, and they're so small, millions of them can fit in a single cell in your body. So they can easily go into the blood-brain barrier in these little balls. But when they get up in there, they go pow like this, huge, and then they do their job. Now they either turn into something that breaks stuff and just unassembles things, or they put into something that assembles stuff. Two different types of bacteria, or virus maybe, but they create these two different things. Now, this is sort of new. The virome, they call it. Now, let's just talk about a virus and a bacteria. I see them doing basically the same. As far as I can see, they do the same thing. All right, like I said, I'm going to stumble through this with you. We know that the bacteria create ribosomes. We know that the ribosomes are these little balls, and when they get a trigger, they just pop open, whatever the trigger is to set them off, and then they turn into these huge enzymes. So far, we got the bacteria creating the enzymes, and they also create proteins. These are the building blocks. These are the breakers. All right. Now, there's two different things that happen. Down here, they're showing that these ribosomes can actually reprogram your DNA and make you basically immune from diseases. I believe that's what they're showing here. But you also have other bacteria, like this bacteria, that's squirting out things to break down food and do everything else. It's not affecting your DNA. So there's two different styles to me of bacteria. One is an invasive style of ribosome that goes after your DNA, and one of them is just does maintenance throughout your body. So they do two different things. They can create immunities, or they can go and break things up and build things up for enzymes and proteins. Let's take a look at this process right here, because this confused me, and it still does kind of confuse me. But I'll show it to you, and maybe it'll confuse you. Who knows? All right, this is the way they portray it, and I'm not exactly sure why they show it this like coming straight through the ribosome, but let's get to that in a second. These are all the amino acids, and they come through these chains. I think I showed you before, and they can be short or they can be long. It depends on what they're going to do. And they are actually, in my mind, they're balled up inside the ribosome. And they float around in this ribosome. All right, so they don't transfer any RNA until they get to where they're going. And then they hit a trigger and poof, they squirt out all this here. So let's see that actually happening. All right, so this, I'm not sure about this, but they do transfer the RNA sequence, which is the polypeptide chain, from the ribosome into the RNA. It goes out and programs the DNA. That, I believe, does happen. And that's the way they claim it happens. And here it is supposedly happening. Now, let's just take the bacteria. As I said before, they all have their own DNA. You see it there? They all have their own DNA. And then it creates that little tiny ball, which is a ribosome. All right? And by actually squirting out that polypeptide chain, which does create the ribosome. See this little foggy area here? That's the ribosome. All right, so we got it squirting out and programming this ribosome, and then the ribosome in my mind goes out and gets triggered somewhere. Now normally for food and all that stuff to break it down. However, they're showing it squirting out here messenger RNA. Looks like it's just going right through the body. I would have thought it would have to snuggle up to the DNA and push it right in, but they're showing it drifting around. I don't know if that's true or not. And then they're showing the DNA being spliced open. And to splice it open, you need another enzyme. These are enzymes. Whenever you see ACE on the end of something, basically it's an enzyme. This is where you have transferase, where it's transferring. That's what the cloud here is. And this is polymerase. And that is where that splice can open up. Okay, it's an enzyme, and then squirts this RNA in there. And I would say it changes. You see, it's going to change something, break something off, and replace it with something else. I don't know. And it ends up making you immune. But that's how you do it. In your DNA, apparently, all I can do is go by what they say, and it looks like it's pretty good information. Your DNA picks up on this new delivered message and says, hey, here's what the guy looks like. If he comes around, send out this, this, and this. All right, now, the viruses may have to go program new bacteria and say, okay, we got new stuff coming in we never saw it before. Go get something and we'll program it. All right, as I showed earlier, there does appear to be a database that is free to use, but somebody's going to have to compile it into an orderly fashion that we can use it and then associate it to all the different symptoms, the age and where they live and what kind of water they drink, and all kinds of things, so that we can see what affects what kind of bacteria affects what kind of condition, if it does. And I think it will. So we need a baseline of all the standard bacteria that would be a standard model. And if you're way up high here or way down low there, there's probably going to be a reason for it, and there's probably going to be a result. So we need, for now, to start, we just fecal swabs. They're cheap and you can get them, and the results could be very, very good to find out what bacteria you don't have or do have too much of. Now, I did this for the veterans, about the people that were at Pearl Harbor and they got destroyed because they put jet fuel in the water by accident. I mean, they didn't do it on purpose, obviously, but then they lied about it and they let these people ingest it. And now I believe their bacteria is completely destroyed. And they immediately had exactly the symptoms. And guess what? From drinking the water, their lungs were the first thing to go. They start coughing, and can't stop. And that's because it killed the bacteria that's in your body. And specifically, it has a lot to do with hydrocarbon and the going up into the lungs and destroying that particular membrane or the bacteria that's up in there. And you just can't get cleared. And so you need possibly a new set of bacteria. That's what I'm hoping for. And I think the veterans need to stand up and say, we want a database of the bacteria and see if they fit the database or not. And if they don't, we want some new bacteria. That's all. Simple as that. If they fit the database, well, forget about it. It didn't work. But that's not a big deal because we need a database anyway, first of all, which they do have. And why it's not being used, I have no idea. I had no idea it existed. So anyway, that's the deal. All right, I love you all. We need to get this done. Somehow we need to get a database of this and make it available to people to just do a fecal swab for very little money and get it analyzed. The problem is, I think you got to do it within 24 hours, they say. Or you might be able to put it on ice and send it in or something like that. But you need to keep it cold and quickly, because the bacteria, I guess, they say it can get damaged. Well, that could very well be. Anyway, please, somebody step up and get this done. You know, I might have missed something here. I've been screaming that there's no database for bacteria. Maybe I missed this. This goes back to 2022. It says MBodyMap is a curated database for microbes across the human body and their associations with health and disease. Wow. Its primary aim is to promote the reusability of human-associated metagenomic data and assist with the identification of disease-associated microbes. By consistently annotating the microbial contents of collected samples using state-of-the-art tool sets and manually curating the metadata of corresponding human hosts. This map organizes collected samples based on their association with human diseases and body sites to enable cross-dataset integration and comparison. Wow, I never knew anything about this. All I can say is, wow. They're talking about all these different places, I think 22, from within 22 human body sites across 136 projects. They did all this to find out what the bacteria are living where in the body. This is fabulous if they've got a good database on this. I never heard a word about this. Microbes inhabit almost all the human body parts and play critical roles in human health and disease. They inhabit every single body part. The human microbiota is located primarily in the gut. That's where I was working on and using fecal samples. They're doing something a little different. They're using every sample from everywhere. So I'm saying the human microbiota located primarily in the gut, where the numbers and diversity from the stomach to the colon multiply continuously, all kinds of different variations. However, other anatomical parts, including lungs, skin, vagina, eyes, placenta, ears, mouth, nasal compartments, harbor microbiomes. The microbiome's composition varies depending on the anatomy, obviously between the intestines and lungs. Not only that, it varies depending upon the kidneys and the liver and the lungs and the coatings of the membranes. That's where your microbes primarily are living in these membranes. And each one of them, that's why you get cancer, and every single organ has its own type of cancer, because you have your own type of bacteria living there, and if that particular one dies in that liver protection zone, well, you get liver cancer, whatever it is. But they all have their own biotic protectors. All right, so let's just continue on here. The microbiome changes quite a bit between individuals, and even over time your microbiome changes. It can be altered by dietary changes, including use of probiotics, which is good for you, the use of antibiotics, which is not good for you, other drugs, your age, or diseases that have infected and killed your bacteria, other factors and is also dynamic. For instance, the human skin microbiome is highly personalized. Everybody's got a different type of skin biome, depending upon multiple factors: how you wash, and how you clean, and how you do this, how you do that, such as body site, age, gender, and lifestyle elements. In addition to individual microbes, which are known pathogenic bacteria, they claim it, changes in the composition of microbes causing dysbiosis are increasingly observed in many diseases. They finally started looking at it. Colorectal cancer, type 2 diabetes, inflammation bowel disease, Therefore, the importance of maintaining a healthy microbiome has garnered attention over the years. Although the exact definition of healthy microbiota remains to be provided, they still don't know what is healthy. This increased attention has seen probiotics, prebiotics, and symbiotics developed and used to intervene in microbial dysbiosis and restore healthy microbiota in cases of numerous diseases. numerous diseases, they're using the same thing, which is the microbiota. Now, there's something called fecal matter transplant where you get a good set of bacteria from a good, healthy donor. And they can mix the donors now, I believe, and you get like 4 or 5 donors at one shot, and it covers all your bacteria, and you're good to go again. You know, that's what pretty much is what they're saying now. And it's all about this dysbiosis. And this is all pretty new stuff. This is all pretty new stuff. I think this goes to 2022. And in the medical industry, and it's in industry, trust me, it goes slowly. As long as everything just keeps going along the way it's going along, don't take any chances. Don't step out of the bounds. If you get sued, you're going to get sued. Be careful. Don't say anything. Don't, just do exactly what everybody else does. It's not getting anywhere. All right, they're saying all this stuff is free. It's free for anybody to use. And all data are freely accessible to all academic users. What we need to do is get some academic people on this database and create the database using all this stuff. And again, this is way outside of my pay grade. I can do none of this. I'm a moron at this stuff. But if it's they have all this stuff, including 14,000 metagenomes and 48,000 amplicons, and I mean, what it all means, I'm not exactly sure. But if this is truly a database of bacteria that are in the human body, and we can put it in a regular database, and then we can have somebody doing fecal swabs and measure that against this database. That's the bottom line. If we can measure somebody else's bacteria against the database that they have already assembled, it says, and it's free. But somebody's got to do it. Now, there's going to obviously be some money needed to hire somebody or have people do this. I don't have any money. I don't want any money. I can't do it. But we need to have somebody do this. You get all kind of funding from all these people. That's wonderful. But to put it in a database where we can actually use it to go against it with some test kit. And I was looking up fecal test kits, and they have a whole batch of them. And they use them for colonoscopies and stuff. But if they're taking fecal swabs and they have all these test kits available, I don't see why we can't get one and then just make it and see what it breaks down to against the database. That's exactly what we need to do. If that can be done, and sounds like they already got the database. I didn't know about this. So that's one step closer. That's pretty nice. All right, so what do we need? If we have a database and somebody has it and we can input our data into that, how do we get that data to input into it? Well, obviously we're going to have to collect a stool sample. And they have tons of them available. And how would we get that stool sample analyzed to see what that actually contains for bacteria? That's the piece we're missing right at the moment. Because if we can fill that in, we can collect the stool, we can get it analyzed, compare it to the database, then we got something to go by. Is this guy high or low on what? Right? Because bacteria are going to make the enzymes do the chemistry. You see this? They actually do these stool cultures. A stool bacterial analysis, known as a stool culture, looks for bacteria, viruses, and other germs in your stool to help diagnose and treat a digestive tract infection. It's not just a general overall look at enzymes and bacteria and germs and viruses. They're targeting certain ones that probably have made you sick. And they give it certain nutrients to encourage the growth of certain bacteria. So they're not doing a complete analysis of your total bacteria, but they are doing a partial, looking for this, and the other thing. What we need to do is be able to give them a stool sample and say, how does this match up to the database of everybody else? And give me my numbers compared to theirs. Simple as that. All right, you see this? This is microbiological stool analysis by PCR. Now, PCR consists in quickly identifying on a small sample of stool the presence of more than 20 microorganisms from diarrhea and so forth. Now, and then they go down, they can also identify these viruses and bacterias and parasites and so forth. Within a few hours, really, this approach allows the following microorganisms to be identified in a few hours on a single sample. Now, why can't we do all the bacteria and the same thing using AI or whatever, some new technique that just scans the thing and does it instantly? My tests were PCR done on my mudfossils. All right, these were my DNA tests I had done in 2015. And what he did was he targeted a group of sequences of DNA to see if they were there in these samples using certain primers. And he explained the whole thing. Very, very, very accomplished guy. And it came back that all three of them were human. And two of them had very dense DNA. It goes through a whole bunch of stuff down here, da, 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 da. All the tests were negative for negative controls. These were the elutions and the different stuff. But excellent quality DNA sequence obtained from the 36-inch sample and the lung. And here's what they found was these are the two sequences. Mitochondrial B gene and D loop region. All right, now, this is what they're looking for, is these CTAGs. These are the DNA codes that say I'm a female, human, modern Homo sapien. Just like typing it off, that's basically what it says. And this was very well done. And now this was a long time ago, before they had whatever kind of they're doing now. This was, well, almost 10 years ago. So it was back in 6 of 2015. And as far as I know, this is the first that was ever done. Absolutely for certain, the first one was ever done for ancient human and giants, which was what it was. Just what it was. Now, I could be wrong about this, but I don't think I am. Now, I say the viruses are programmers. So they go in and program the machinery that's already there. What machinery? I say it's the bacteria. They say it's the cellular mechanism. Well, I'll tell you what. The cellular mechanism is driven by enzymes created by the bacteria. So instead of producing the correct enzymes, which are the ribosomes, they produce the new ones. They squirt them out the same way, only they're viruses. Now, here's what it says. The virus will enter the host or invade the host cell. So they say it's going into the cell. That's fine because the bacteria are living inside the cells. They're living in the extra cellular stuff and all that. So once it gets in, it starts making copies of itself. It takes over the mechanism. All right? It uses all the cellular mechanisms and energy to make copies of itself. The cells are injured by doing this. They come in and break them up a little bit, and they get weak, and eventually they'll be destroyed because the virus reproduction just kills the cell. Is it just a normal cell of the body, or is it a cell which is basically a bacteria? That's what I'd like to know. Now, don't forget, the bacteria live inside of our human cells as well. They're the factories that are producing all of the enzymes. And what happens is those viruses get inside our human cells and take over the bacteria and start producing viruses out of that bacteria, which is, they're calling it the cellular mechanism. Well, the cellular mechanism is bacteria. All right, pay close attention to this. Ribosomes, as I told you, you could have millions, up to 10 million of them in a single cell, in one cell in your body, 10 million. They're 25 nanometers in diameter. Viruses are in that same range. They can even be smaller, about 20 for some viruses, all the way up to 100 nanometers, which is not big. That's only 4 times bigger than this. And that's so tiny, millions can fit in a cell. So these are also extremely tiny, and they squiggle into the cell through loose junctions. Let me explain to you that. All right, you see this? That's a cell membrane right there. That whole thing right around there is a cell membrane. That's this. It's got 2 layers. They're phospholipids. It means phosphorus, basically phosphorus, and fatty layer in between. The only thing that's supposed to get into this cell, the only thing, is through these channels. They call them ion channels and the protein channels, whatever you want to call them. But they come through, and that's the only thing supposed to be allowed in, and the only thing that's supposed to be allowed out is what they want to expel with bacteria or whatever. But this is the layer that protects you. All right. Now, how do the viruses get in? Don't forget, they're inside of this. There can be 10 million ribosomes. Now, all you need is a couple little viruses to get in here and take over some of these ribosomes, and then they start to take over and spit out more and more of themselves, which take over more and more and more, and you get invaded pretty quickly, unless you can fight back. And that's basically what's called the viral load. You hit a certain point, and you're just going downhill, more and more and more infections. It's like a cascade. So you get a couple through here, yeah, they could probably handle it, but best not to get them through here. But once they're through, you better have some bacteria in here and some ribosomes in here that can attack those things. When they're new viruses, no, you never saw them before, you never had them before. And that's what happens, these seasonal flus and so forth. They're new, little twist to it. Your body's never seen them before. It doesn't go and attack them until it feels it's really being attacked. Then it fights back. By literally now, I think it tells a virus to create a program to go kill that thing. It has to figure out what it is, what's its chemical signature. So what will happen is the virus will go out and it'll look at it and say, oop, there's a spot where we can get it, there's a spot over there, a spot over there. It goes back and it tells the bacteria to program to do whatever it wants, and then that new ribosome can go out and attack that new virus. I mean, the new virus or whatever it is it's got to attack. Because something new comes into your body you never saw before. Somehow you have to deal with it. So you have to reprogram your bacteria to deal with it. And I say the virus is the programmer. Now it all makes sense because it takes over your body and reprograms your body. They know that. It takes over the mechanism of the cell. The mechanism of the cell is bacteria. And then it creates another tiny little molecule, which is its ribosome, basically, but it's called a virus. And then that goes floating through your system, and it's small enough to go through the blood-brain barrier, I'm sure. 20 nanometers is smaller than the ribosomes, and there's 10 million of them in a cell sometimes. So you gotta start thinking on different scales than you ever thought of before. But if you get broken through up in here, you're in trouble. And especially if there's enough of them and you can't fight back. So first of all, let's put it this way. If there's enough of them, even if you got something that can fight back, you may get overwhelmed. If there's just a few of them, you better have something that can fight back, and then you can usually wipe them out. If you don't have anything that can fight back, only a few of them can come in, and once they take over, they start making the other ones do the same thing that they're doing. So now you're in trouble. All right, so you just saw the membrane. This is the cell in its magnificentness. Of course, that's the membrane that surrounds it. Now, here you got ribosomes. They're all over the place. And like I say, there can be up to 10 million of them in here. And you got all kinds of Golgi apparatus and all kinds of things that secretory vessels. They secrete enzymes and juices. You know, this is just one single cell. One single cell. Now, the ribosome, the virus breaks through somehow through the plasma membrane. It's the only way I can think that gets in there. Through a loose junction. I'll show you the junction in a second. But once it is inside there, now it's all over, unless you can fight back against it. So normally you're going to be invaded by viruses that you have defenses against. You kill them, you never even knew it happened. And hopefully that's what these flu, inoculations are about, is to try to give you those kind of proteins in your body to fight back when it sees this particular chemical signature. Because its chemical signature sees it and says, oop, that's it, wham, and it's all over. Normally your body will do that. But sometimes your body can't do that, and then it's all over for your body. All right, so I say, where is the weak spot? Well, here's the deal. This top here is your skin, let's say, or the top of your fascia. And down here, this is the coating, the membrane, basically. All right. Now, there's 3 junctions. There's the top junction is a tight junction. It's supposed to seal here. They could twist around and move around, all that, but it should never break open and allow stuff down. You got another one down at adheron junction, so that they really stick together pretty good. This is the gluey stuff, and it's floppy because it can move around. And this layer right in between here has the enzymes in it. It apparently has the catalysts, and they're the first line of defense. So if it breaks through here, this is where your enzymes and catalysts are going to live primarily. Now, after you get past this tight junction area, you go into what's called the adherons junction, and these just keep it adhered tight. All right, and then you get down to the bottom layer, and it's called the I'm sorry, I'm amazed they don't have endosomes. Endosomes, I think it is. Down at the very bottom. I don't know why they don't have that labeled. Interesting. But anyway, these are the things that are in the layer of skin, basically. And then you get down to the basal lamina, which is the basement layer, where this outer layer adheres to. So basically this is the skin. Think of it that way. Oh, okay, this was my mistake. These are the desmosomes right here. See them over here? Desmosomes? Denominosomes. I can't really pronounce it that well. But that's these guys right here. Same thing here. But then you have the adherons junction, which is up above. Now, I'm not sure what these do, to be perfectly honest. I don't know about the gap junctions either. But there's a whole bunch of junctions that hold these tight. You can't get through there. If you get through there, you're in trouble. And a lot of people have loose junctions because the gooey substances that are supposed to be in here, the enzymes probably are not working to create those substances. Or you're not eating the right food to create the substance. Or something's going wrong. But we should be able to find out if you have the bacteria in your body to create the substance. Because you need to have the enzymes to create the substance. You don't have the bacteria, you don't have the enzymes. You don't have the enzymes, I don't care what happens, you're not going to get the substance. So it all starts with the bacteria. And because there's so many different strains, we need somehow to get a database that says this is pretty much the standard. And if everybody has pretty much this level of bacteria in their body and all of these different ranges, because every one of them has a job to do. And so, because when you break down your food, it's not just one click and it breaks it down. It uses probably 500 enzymes. And they all break in half-lives. It goes bang, and then grabs another one, and they work together now. Now I'm starting to pretty much understand the consequences of these half-lives. They're so fast that it breaks down to chemistry, and then it breaks another one, and then it ends up being what you wanted in the first place. It may take minutes, it may take an hour, but there's going to be so much chemical activity, and you need all of those bacteria creating all of those enzymes. And then you'll have, this will be all sealed. You'll have plenty of protection up here in your enzymes, and your catalysts will be there waiting to protect you. The gut junctions will be nice and tight. Everything will be working fine. But if you don't have the enzymes, you're done. And if you don't have the bacteria, you don't have the enzymes. This is how they image this human cell. I don't care about this stuff. This is a sample chart, but here's the cell. Here's how they made all these little whatever they do for the different colors and so forth. They break them down into whatever this is. And then here's their end rendition of what a cell looks like if you've cut it across, using whatever they used. And I don't care about that. But you see how complicated this is? I'm going to show you another one that was sent to me. I don't know where it came from. I don't know if it's accurate or not, but it certainly could be. All right, I know this looks insane. It's going to be crazy, and it's probably not accurate. However, take this into account. There's all kinds of organelles in here and little bits of mechanisms that are working inside there. Golgi apparatus and the whole 9 yards. Now, these are the exits from the cell out, I believe, into the fluid-filled highway. And these, I would believe, are the membranes that surround the whole thing. Don't forget, you can have 10 million of these little ribosomes inside of here. So this is not out of the range of possibilities, I wouldn't say, but I don't know. I mean, it seems pretty exotic. I certainly can agree with that. Now, is this the fluid-filled highway? I think so. They are supposed to create enzymes and do all kinds of things in here and have the ribosomes, little ribosomes floating around everywhere. 10 million of them. Now, all of these little things that are attached to these cellular structures are the things that take in and release chemistry so that you can live. That's what they do. Now, is this true? Is this correct? I don't know. I do know things are constructed like this, like these little things like that. There's all kinds of little chains of molecules throughout your body, and they're in chains of polypeptides. Now, these, I think, are going to be the little channels that run out into the fluid-filled highway. So the bacteria, or whatever's going on in here, has to be able to get out of here. And there's some kind of a filtration system and so forth to get out of here. It looks to me. I don't know. And again, this could be just totally, somebody's fantasy. I have no idea. But I can tell you what, there's a lot of chemistry going on in every single cell. And again, 10 million ribosomes. Just think of that. That's just beyond belief.